Hello, I'm Nicholas Tatonetti, Associate Professor of Biomedical Informatics at Columbia University, and I like to think about how to use publicly available massive data sets to make scientific discoveries. Today I'm going to be talking about how we use data mining, integrative data mining approach to identify a novel and unexpected and dangerous drug-drug interaction. Acquired long QT syndrome is a dangerous drug-induced ventricular tachycardia, a heart arrhythmia that can be caused when patients take certain medications. Now, this is so important that the FDA mandates that every single drug be tested for this effect before it even enters clinical trials. If there's any identification of a signal against this important cardiac ion channel, which is responsible for this arrhythmia, then that drug is usually dead in the water and doesn't continue on. But there's no such mandate for drug-drug interactions. In fact, we must rely on data mining of observational data sets, complete with all of their missingness and bias limitations to identify these drug-drug interactions. But how do we know that we have a good hypothesis, even if we find a signal? There's so many false positives in this data set. So we take an integrative data mining strategy. We don't rely on just one database with one analysis. We bring together multiple orthogonal databases and techniques to improve the hypothesis generation process. In fact, the more different the databases are and the more different the algorithms are, the better we feel about the hypotheses that are being generated. In this analysis today, I'm going to show you how we use the FDA's Adverse Event Reporting System, a side effect database, to identify putative drug-drug interactions, how we integrated a completely orthogonal database, the electronic health records at Columbia University, to corroborate those hypotheses, and then how we validated that hypothesis using a prospective experiment. We invented this algorithm called Latent Signal Detection to look for drug-drug interactions. Drug-drug interactions are not typically reported in these databases directly. Instead, there's lots of different individual drugs reported with their side effects. So we have to figure a way to infer that these drug-drug interactions are occurring. And in fact, we invented this algorithm is inspired by how scientists hunt for planets in in the universe outside of our solar system. And in fact, how it works is by inferring the presence of the drug-drug interaction by the observable effects that we can see. So in fact, what we do is we assume there's some unmeasured severe effect lying underneath some level of detection. And we hypothesize that this unmeasured severe effect is eliciting certain observable, more common minor effects. If we can identify those minor effects, which we are going to using supervised machine learning, then we can put them into a profile and use that profile to mine for drug-drug interactions. So that's exactly what we do for acquired long QT syndrome. Now, we don't know that this drug-drug interaction is happening directly, but through the presence of more minor observable effects, we can infer its likelihood. We apply this algorithm across the entire database that the FDA makes publicly available, and we rank all possible drug pairs for their potential for this drug-drug interaction, how much they look like they're causing acquired long QT syndrome. Our top prediction was between ceftriaxone and lansoprazole. Now, ceftriaxone is a common inpatient antibiotic, a first-line antibiotic here at our hospital at Columbia University. And lansoprazole is a proton pump inhibitor, one of the most commonly taken drugs around the world. In fact, it's sold over the counter as Prevacid. Now, interactions between these two drugs, especially causing this heart arrhythmia, could have dangerous outcomes. These are the profile that the supervised machine learning algorithm used to identify these effects. We asked the algorithm to do one extra step, which is to predict another cephalosporin antibiotic, cefuroxime, and lansoprazole to predict to identify another combination that would not have this effect. So this profile below is not indicative of this drug-drug interaction. Then what we do is we go to the electronic health records, a separate database using a separate type of an analytic technique, but we ask the same question. Is ceftriaxone and lansoprazole prolonging the QT interval? And is cefuroxime and lansoprazole prolonging the QT interval? And what we found is that patients that were exposed to both of these drugs simultaneously had 10 millisecond longer QT intervals than patients who were exposed to only one of the drugs, a significant sign of this tachycardia occurring. We did not see that effect in the predicted negative control. Now it's time to go to prospective experiments. In this experiment, we take cells that are overexpressing this important ion channel that governs whether or not a cell is going to have this arrhythmia effect. And we have cells that we treat with just control. We have cells that we treat with ceftriaxone alone. We have cells we treat with lansoprazole alone. And then, of course, we have cells that we treat with both ceftriaxone and lansoprazole. These are electro traces of the current across the membrane of these cells. And what you can see here at the top is in ceftriaxone alone, there is no difference as you increase the dose on the activity of this ion channel. But as you increase the dose of ceftriaxone in the presence of lansoprazole, you see this dose-dependent effect. And in fact, you do not see this for the predicted control. If you aggregate this over many cells, you see this beautiful dose-dependent response curve showing the effect of this combination that you do not see from the predicted negative control. Now, if you plug this back into a model and predict how this would affect humans with this type of experimental result, you would actually expect a 10 millisecond longer QT interval in patients exposed to this combination of drugs, exactly what we saw in the electronic health records. So in this study, I've shown you how we can use observational data sets. We can mine them for hypotheses, that we don't trust those hypotheses outright, that we corroborate them with independent analyses of electronic health records, and then finally that we validate them prospectively using a completely different technique to prove that this interaction is actually happening.